morning or good afternoon, depending on where you are. Welcome to EHE Community Connections. Thank you for taking the time to be here today. My name is Maggie Cameron. I'm the Director of Development and Communications at the EHE Foundation, and I'm also a patient. Before we get started, I need to share a few housekeeping items. We're going to ask that you put any questions you have in the chat. You can find that down at the bottom of the screen along the toolbar. Also, if you don't mind, please keep your audio muted at all times during the presentation. You're welcome to keep your camera on. However, if you have a lot of movement around you or you're moving around, please turn your camera off so as not to distract the presenters. Again, we want to remind you and encourage you to submit any questions you have by using the chat at the bottom of your screen. We want to also remind all participants that the EHE Foundation does not provide medical advice. The information provided today is for your information, and we encourage you to take all questions to your healthcare provider. Okay, let's get started. With us today is a familiar face, Denise Robinson. She is our Director of Research. Take it away, Denise.
Thank you, Maggie. Thanks, everybody. I appreciate your joining us for Science Saturday. It's good to see you. Thanks for being here. So I want to talk to you today and give you some updates on what's going on in EHE research. I want to go through how you are powering EHE research, and then we're going to leave time for questions and discussion at the end. So as Maggie said, feel free to pop your questions into the chat anytime, and we'll get to those at the end of the presentation. So I want to start first with our why. This is why we get up and do what we do every day. It's why we're here, our mission and our vision. Our mission is to find treatments and a cure for the rare cancer, EHE, by advancing research and driving collaboration between patients, researchers, and clinicians. And we feel like it is important that patients are at the table with researchers and clinicians in order to really advance research. Our vision is a world where EHE is easily diagnosed and treatable. And we know that today that is not the situation. And that's why we're here, to change that and make sure that people can be diagnosed and treated who have EHE. So I want to start by talking about the drug development process and where we are working to advance knowledge to find treatments for EHE. So we know, I've listed some key accomplishments here. There have been a number of accomplishments in drug development, and these are just a few. The drug development, the timeline is quite long. It's 10 to 15 years on average to develop a drug. It takes hundreds of millions of dollars. And already this community is coming together, and scientists, leading scientists, have made these key accomplishments and enabled further research. So if we look back to 2011, Munir Tanas published the disease-defining gene fusion, WWTR1 or TAZ CAMPTA1 fusion. We know that about 90% of people diagnosed with EHE have the TAZ CAMPTA1 fusion. And then we know now that the other 10% had the YAP1 TFE fusion. This was an early discovery that enabled a lot of research. Further, we have a genetically engineered mouse model and cell lines that have been developed from the Rubin lab. This model of EHE has been very instrumental in further discoveries in EHE. And then we've more recently learned about the TAZ-CAMPTA fusion and interaction, and that has led to clinical development. We've come a long way from this basic research, and now in this seed inhibition, we have clinical trials ongoing now, early phase clinical trials in EHE. So a lot of progress has been made along this drug discovery timeline. At the EHE Foundation, our research grants program really seeks to help all aspects, all parts of the timeline. So far, our work, our money have been mostly focused in this research and early discovery area and preclinical translational science, but really we want to be able to support all areas of drug development. EHE biobanks are very, very important, particularly in this basic research and early discovery laboratory, uncovering elements of EHE biology, model development is critical for drug development. And then there are some foundational studies, so patient registries, the EHE Global Patient Registry and other registries, the prospective observational studies, and retrospective studies. So these studies and registries are all very important for generating data. We need that real world evidence and data of what is going on in patients in a clinical setting and reported by patients. And all of this data, all of these things together really seek to advance the drug development timeline. And all of this is progress that's going on in this community. Let's talk a little bit about where progress is being made in EHE research. And I'm going to shout out here to several very significant collaborative EHE centers. And I also first want to make an apology if you are a researcher or a clinician and your institution's logo is not represented on this page. This is by no means meant to be an all-inclusive list of everyone who's doing EHE research. I've highlighted these because these are really significant, multi-pronged, multinational, multi-year, mostly research efforts that have been going on, and these are excellence, centers of EHE excellence overall. I'll start up in Canada at Princess Margaret, led by Dr. Razak. ProCare is a multi-year, multi-pronged project that seeks to go from basic science through translational science. It spans Canada, patient-powered research truly through generous donation of dollars, enabling this to go on in Canada, and it's significant collaboration ongoing there. Of course, you all are highly familiar with Dr. Brian Rubin and his lab at the Cleveland Clinic. Numbers of publications and findings have come out of the Cleveland Clinic, much of which has enabled all of this other research to happen. And we could not be more grateful to Dr. Rubin and his team there. I'll jump left to the University of Iowa, Dr. Munir Tanas, who I just mentioned in the disease-defining gene fusion. Dr. Tanas was early on in the Cleveland Clinic. We are glad to be able to partner and support Dr. Tanas now, and he is a world-renowned researcher in his own right in EHE and relevant science. We're glad to work with him. At Albany Medical College in New York, this is another lab that was seed funded by this community with small grants that have enabled this to really become an excellent EHE laboratory. This is also born out of collaboration with the Rubin Lab and significant collaboration going on there. And then this wishbone icon here, if you're not familiar with this logo, this is Memorial Sloan Memorial Sloan Kettering Cancer Center out of New York. This is a center of EHE clinical care excellence led by Drs. Tapp and Dr. Christina Antonescu in New York. Significant clinical information and research coming out of this institution. And then I'll jump across the pond here to the Royal Marsden, led by Professor Robin Jones, the ICR, the Institute of Cancer Research, with Professor Paul Huang, and then Dr. Sylvia Stacchiati and her colleagues at the Institute in Milan. And this is a multinational, multi-institutional collaboration. We call it the European collaboration that is ongoing. Again, significant effort going on in these labs to advance EHE research. And I have to give a shout out to all of our colleagues in the EHE group, Fiona Ross in Canada, who works through the Sarcoma Foundation of Canada, the EHE Rare Cancer Charity UK, led by Hugh Leonard. The EHE Rare Cancer Foundation Australia is an important entity in that part of the world. And our newest group on the block is EHE Italia. So all of us together refer to ourselves as the EHE group, and we work collaboratively. We work to pool our funds, pool our resources and our talents, support all of these and many other centers of EHE excellence around the world.
Denise, if I could just interrupt for a moment, would you mind turning off your camera? I want to see if that improves your audio just a little bit. It's just a little bit in and out.
Yep.
Thank you.
There we go. Let's see if that helps. Okay.
I think that's better. Okay.
Okay, great. Thank you. So let's talk about where we're making significant progress in EHE models. So we talked about this a little bit last year in Science Saturday, and we've made more progress since then. I want to call your attention to just two types of models here, and there are other types of model systems that can be used to study a disease and EHE, but I'm going to talk about these two here. So the first one is a PDX or patient-derived xenograft model. You'll hear us talk about this. So this is where a tumor is taken out of your body. Maybe you have a liver resection or a transplant or other type of surgery in any body part. And that tumor tissue is put in a little dish. It is put in some media that makes it happy, it makes it want to continue to grow and live. And then that tissue is soon implanted into a mouse. And these are scientific mice. They're specifically made for drug testing, drug screening, drug development processes. And if that tumor grows and lives and expands in that mouse, then that tumor is passaged into more mice. And again, if those tumors in those mice continue to form EHE cells and they continue to grow in these mice, then those tumors are passaged and more mice. And this becomes a colony. And now we have a PDX model, a patient-derived xenograft model, with which to study EHE. And this is important because we do need to be able to study the disease and drug interaction or activity in an animal system. And then another type of disease model that we're studying and trying to form are cell lines. So EHE cell lines or cell cultures are made in a dish. This is where a tissue, a tumor tissue is taken out of a liver. It could be taken out of any part. We're just showing a piece of a liver here, and it would be taken out and put into a dish again with some growth media to make it happy and make those cells want to grow and expand. And if the EHE tissue and tumor cells continue to grow and expand, then they are passaged into more plates. And again, they continue to grow and expand and they'll passage again. And this is how a cell line, this is a very lay and simple explanation of developing a cell line from taking tissue from a living body and putting it in a dish and letting those tumor cells grow and expand so that we can study those. So we know already, we talked about this last year, it's been well reported that a genetically engineered mouse model has been developed at the Rubin Lab. And from that, we have cell lines with which to work and further study disease, both in a cell line, but also in that mouse model. We have a patient-derived xenograft model, a PDX, that was made at the INT Milan. So that has been an important model for that science and that team to use. Most recently, and the thing that we're very excited to share, is that at the Fred Hutch in Seattle, Washington, there is an EHE human cell line. So if this line is fully characterized, this would be the first human cell line developed. There is further work to be done to characterize that line, and we look forward to sharing that information with you. But that's really exciting development in science, an EHE cell line in EHE. So in models and development, there is another version of a genetically engineered mouse. So the YAP-TFE3 form of EHE. That genetically engineered mouse model, or GEM, is still in development. A zebrafish model has been in development at the University of Sheffield. Again, this is a different model system by which we could study EHE in a different animal. There are EHE cell cultures in development at the University of Iowa. We look forward to reporting out on those in the coming year. And then another patient-derived xenograft in the plan for the ProCare team up at Princess Margaret in Canada. And then lastly, another cell line, and this is a shorter cell line in development at the Boehm Lab at MIT. This would be subjected to a CRISPR screen, looking at drug dependencies, and it has a very specific use and intent. But again, we're so glad to report out that these models are in development. We anticipate that there might be other models in development around the world that we don't yet know about, but these are ones where we are partnering to bring those models forward. Stay tuned for that for further progress in this area. So another area of progress that I want to talk about, and this also relies on models for parts of drug repurposing. So drug repurposing for EHE, if you don't know this term, it aims to identify existing FDA or other agency approved drugs that could work and be effective for the treatment of EHE. So essentially, this is using drugs that have already been well tested to be safe and effective in humans for other diseases. So it may be for a cancer, it may be for another disease altogether. These could be drugs that you could purchase over the counter. So you could walk into your pharmacy and you could buy them without a prescription, or they could be drugs that could be prescribed by your doctor to treat EHE. And usually they're prescribed or they're indicated to treat another disease. So already we know that many EHE patients are being prescribed or are using what we call off-label drugs to treat EHE, and with some success and not. It's well reported by patients and in some smaller case studies and reports from clinicians that drugs are used, and we hear reports of good outcomes or not good outcomes. But what we don't have is a consistent use of those drugs in large data sets. So right now we have inconsistent use of drugs because there isn't a lot of data, and we don't really know, we don't have well studies to say how those drugs have worked in EHE patients specifically. We may know how those drugs work in other people in other diseases, but not specifically in EHE. And what that leaves us with, because we don't have good data nor good use, is an inequality in patient access to drugs in different regions. So, for example, I'll give you, if you live in the United States and you would like to use sirolimus to treat your EHE, your doctor may be able to prescribe that to you relatively easily if that drug is right for you. However, if you live in Europe, for instance, your doctor may not be able to prescribe that drug because that drug is not proven and labeled for EHE specifically. It does not have regulatory approval and it is not labeled to treat EHE. So we have an inequality in patient access by region. So drug repurposing really seeks to generate that data that we need and evidence. And that data and evidence would ultimately be used to seek regulatory approval. So you could take that data and evidence to a European agency or the FDA here in the United States to gain regulatory approval specifically for EHE. That could mean that that drug could have an expanded labeling, so it would be indicated specifically for EHE. Again, no drugs today are indicated specifically for EHE. And ultimately, the goal is to share that data. So if we collect this big data and we collect this evidence, that data should inform your treatment. It should inform your clinician in his or her decision to treat on what drugs have been proven and studied, well studied to be safe and effective. And it should inform you on your care decisions. And again, this is using existing drugs. So this is ideally a shorter pathway to find drugs that could work for EHE than developing a novel or new drug that is not yet approved. There are a number of active projects that are really directly working on drug repurposing, and I just want to do a shout out to a couple of those here. We'll talk about some of these more. Sirolimus for EHE has been a multi-year effort, most significantly led in the EU because of the lack of access due to the labeling. But this is a significant effort. And then also I want to say the Cure Drug Repurposing Collaboratory. So this is a public-private partnership in the United States, and this team works tirelessly to help us find drugs that could be repurposed and effective in sarcomas, specifically EHE. They work also to support other diseases, and mostly they are working on drug repurposing. So these are major collaborations that are going on in this area, and we do look forward to progress here. So the first project that I want to highlight for you is Dr. Ajay Pabati at the Cleveland Clinic. And Dr. Pabadi is working on a project entitled Harnessing FDA-Approved Drugs to Gain Mechanistic Insight into the Regulation of the Taz-Canta Fusion Protein. And that's a mouthful. But essentially, this project builds on data that was generated by Dr. Pabadi and his team. We awarded him at the foundation a 2022 Research Grant Award for a project entitled Repurposing an FDA-Approved Drug for EHE Treatment. So in 2022, that project sought to screen a couple of thousand FDA-approved drugs. Again, these are drugs that are proven to already be safe and effective in humans for other diseases. And he would identify drugs that could regulate the location of Taz-Canta within the cell and cause it to be unstable. The idea is to drive Taz-Canta out of the nucleus of the cell, ultimately destabilizing it, causing it to degrade and disappear. So from that 2022 grant, he generated enough data to submit an application into our 2023 research grant cycle. And from that data, we awarded a 2023 research grant award. And that project aims to further investigate and understand the potential of the drugs that he's identified, again, out of this approximately 2,000 drugs screened, that would regulate Taz-Canta. So we look forward to more data coming out. The 2023 award results in work that happens actually in 2024. But the thing that we're really excited about, we've just learned from Dr. Rubin as the lead PI, that the NIH here in the United States has given a $225,000 grant to further expand the work that we funded through these very modest grant awards to Dr. Pabadi. They generated enough evidence that enabled them to get this additional money. So this is our good money from this community and good work put to good use to get this larger funding. So stay tuned as we look forward to readouts from Dr. Pabadi and Dr. Rubin on this research. So the next team and the next project that I want to highlight here is Dr. John Lamar at Albany Medical College. Many of you are well familiar with Dr. Lamar and his team. They are huge EHE advocates and champions. They are always involved in our community and lend their time and support in many ways. Dr. Lamar has many projects going on in his lab, but the one that I want to talk about today is the use of preclinical EHE models to identify druggable pathways to treat EHE. Again, that is a mouthful, but essentially, Dr. Lamar and his team aim to reveal pathways that can be targeted with existing drugs. So here we are, drug repurposing. We're looking at existing drugs to see which ones of those could eliminate EHE or prevent its growth. And as of his end-of-year report at the end of 23, Dr. Lamar reported that they have established preclinical tumor models of EHE in mice, and those are useful because they can do these small drug screenings and identify compounds that could block EHE cell growth, and that screening serves as a proof of concept that the approach he's using could work on a larger scale. So far, again, his end-of-year report is that they have revealed 17 drugs that could reduce EHE cell viability without impacting normal cells. Again, that is important for toxicity. And 4 of those are in a family of drugs that are proven to be safe and effective in humans. So what we look forward to in this coming year is Dr. Lamar and his team sharing readouts as they continue to investigate mechanisms by which the drugs that they've identified in these family of drugs that can inhibit EHE cell viability. So very, very important work going on there. So please stay tuned as we share more. And the next significant project that I want to talk about is the evaluation of cytokines and hormones as biomarkers of EHE. So this project is led by Dr. Silvia Staghiati and Nadia Zafroni at the INT in Milan. It is a collaboration with Professor Robin Jones at the Royal Marsden in London and Paul Huang at the Institute of Cancer Research, all renowned researchers, clinician scientists. And this is a multinational, multi-institutional, multi-year project that has many aims and objectives. It aims to identify novel prognostic and predictive markers of EHE. Today, there are no biomarkers of EHE, so this is really important to better understand those prognostic and predictive markers. It will better describe the natural history of the disease through observation of patients. It will discover the radiological features of the disease as well. So several of the aims of this project, and again, there are many, one of them is to develop a PDX model and a human cell line. So this has been done. And that model and that cell line have already been useful to this team to evaluate the efficacy of drugs alone and in combination. So you can look at singular drugs, combinations of drugs, including sirolimus for EHE. One of the other aims is to correlate those radiological images to biomarkers or potential biomarkers of EHE, looking at disease progression and drug response according to radiological images. Again, this is a pan-European study. It observes patients. Patients can provide their consent and join the study, and they'll be followed to document the natural history of their disease and looking at any treatments they have and outcomes. Patients can donate blood and tissue samples, and those are informative for that model development as part of this project. So stay attuned. We expect publication and further growth and development from this lab in this research as we go through this year. Another really exciting project, this was just announced and launched to the clinician community in November of last year at CTOS. This is a project, I love the name of it. It's the PUSH project, Pushing Rare Sarcoma Beyond Hope. And EHE is a rare sarcoma. You have to love this name, that we're pushing rare sarcoma beyond hope. This is also championed by Dr. Stacchiati with Dr. Tapp and a number of clinicians around the world. It is a collaboration of advocates, people like us, academic researchers, and regulatory agencies. And this project, it is a platform project which really aims to build essential data with which to advance drug development. And that is going to include drug repurposing, so not just novel compounds, but drugs that are already used off-label to treat EHE. It leverages, importantly, patient-donated data. So your donated data can go into the study through patient registries that you may participate in, prospective studies, donating your clinical data. And ultimately, all of this aims to speed up the process of finding effective treatments. So it's an alternative to industry or pharma-sponsored trials aiming to optimize costs and shorten the process. We do hope that data and the collaboration of this platform will increase the interest of pharma and other funders, because that is important for pushing rare sarcoma beyond hope for EHE. I do want to give a shout out to a couple of institutions on the right here. The Anti-Cancer Fund is a nonprofit institution out of Belgium. They pour significant time, effort, and resources into rare cancers and EHE in this project, as well as other projects, drug repurposing being one of them. And again, I mentioned the Cure Drug Repurposing Collaboratory early. They are a key element of this platform project too, and we thank them for the time and effort that they give to the project and to the EHE community. So stay tuned as we share more on this project to come. Again, it is very new. So let's talk now about how you are powering EHE research. And there are 3 projects that are very near and dear to my heart. I'm so proud to bring these projects forward to you. These are made with patient experts, with clinicians. They're formed for you and they're powered by you. So the first that I'll talk about is the EHE Global Patient Registry, and then the EHE Biobank. And the last project that I want to mention, I'm not going to go into a deep dive on this today, is the XCures Prospective Observational Study. This is a very new project. It is still in a soft launch mode. Ultimately, this gives you a way to participate in research by donating your clinical data. Essentially, that is your medical records. So your clinical data that your doctor records in your medical records, is important to be studied to understand what treatments you've had or not had, your diagnostics that you've had and how that has changed over time and what those outcomes were. And this really complements that you would contribute directly into the patient registry. The patient registry is your story, your experience in your own terms. The observational study is your medical data or data told by your doctor. And those may have different answers. So we would like to have the full picture from both of those perspectives. So stay tuned for more on the XCure study. But first, let's talk about the EHE Global Patient Registry. So this is a natural history study of EHE. Anyone living anywhere in the world can log on to eheregistry.imrare.org to join this initiative. It aims to build knowledge and truly accelerate research. One of the key parts of the registry aims to discover and learn more about treatments that people have been using. And I say treatments, but that includes what we'll say is wait and watch or active surveillance. So if you have not had any treatment for your EHE, your participation in telling us about your experiences and those outcomes is equally as important as those people who have been treated. And another important aspect of the registry seeks to characterize pain in EHE. We know that pain is a component of many presentations of EHE, and it's important that you as a patient tell us, characterize, and describe your pain in these validated instruments and help us understand what pain management tools and techniques and therapies you have used and how those have worked. And again, your pain reporting is maybe different than the pain reporting that could be in your medical records. We know that pain is often underreported in clinical care notes, so it's important that you tell us in your own words. All of this data really aims to support and expedite drug development. So we'll uncover questions, we'll uncover data that will give us a nod to go and explore something further and ultimately improving your care. So through all EHE patients coming together and sharing their experiences, that data, if we pull that all together, would inform clinicians on outcomes and utilization of therapies and techniques to care for EHE patients, and it will inform you in your care journey. So the data that we give back is important to you. So I want to share with you just some of the learnings that we've had so far from the registry. And I am so thrilled to say that we haven't even been open for an entire year yet with the registry. We already have 208 participants from 19 countries. That is absolutely amazing. The registry is actually only available currently in English. However, you can see that people from all over the world have joined in and have taken surveys in the registry. These are very simple surveys that you can answer. And we do know that some people have successfully used Google Translate or other translation systems in order to translate these simple questions, answer their questions, and it has worked. So thank you for everyone who's joined around the world. I'm giving you some simple summaries. Again, this isn't a full data analysis of the data that we have in the registry. These are just some highlights. Symptoms before diagnosis. So when you join the registry, we ask you about how your disease presented when you were diagnosed. And one of the questions surrounding that is, what signs or symptoms might you have experienced that could have been related to your EHE when you were diagnosed? So if you look back in time, what were those things that now you can look at and say, that was probably related to my EHE? The number one reported symptom is pain at tumor site. The second largest one is fatigue. And the third largest one is abdominal pain. These are well reported. This also follows along with literature. But look at the fourth most reported, which is none. We know that EHE is very often incidentally diagnosed. So you didn't go to the doctor for any pain or other issue. Perhaps you had an accident, you had a scan for another reason, and unfortunately it was discovered that there was a tumor or a mass and you were diagnosed with EHE. All of these symptoms, we really want to dig in more and look at the presentation of EHE. Where did EHE arise in the person's body? Was it metastatic at the time of presentation? And understand symptoms and symptoms that occur after diagnosis further. And then over here, I've given you just a very simple summary. So we want to confirm that you have been diagnosed with EHE to be in this registry. We ask you when you were diagnosed. You can see that we have people that have been in the registry. They're newly diagnosed. We have people that are less than 1 year in their diagnosis. But look at this. We have people all the way to 25 and 30 years. So this is really important. These people that are in this longer lived time with EHE. And we encourage you, if you have lived with EHE for quite some time, please go into the registry. Please consider joining and telling us about your presentation when you were diagnosed, how your EHE might have changed over time, and any treatments you've used. Your information is so important to inform newly diagnosed people with EHE and help clinicians and researchers understand this disease. And here I've got a few more graphs and charts. Baseline is what I'm talking about here. And baseline is when you join the registry. So essentially, the day you sign up and you go into the registry to answer your surveys, you're going to answer questions about your diagnosis. And if you were diagnosed, let's say 10 years ago, that's your diagnosis date. And then today, the day that you're joining the registry, is baseline. So 40% of people who joined the registry said local disease at baseline. 60% say that they have metastatic disease when they join the registry. Again, this is baseline, this is when they join. And this tracks right along with published literature on what we see in EHE, which is also important that follows along. 36% of people at baseline said that they've never been treated for their EHE. 64% of people say that they have been treated for EHE. So it's important for us to go into this and say, okay, where were those presentations of disease? How long have those people had their EHE? And what do those treatments look like? What tumors, what presentations of EHE were treated? We look at tumors that were reported at baseline. So again, we're looking at metastatic disease here. Liver and lung and bone are well reported to be the most prominent sites of EHE presentation. 72% of people who joined the registry, and this is a simple summary. I've only categorized the combined metastatic locations. These are body parts or disease organs that are impacted. So 72% of people have liver and lung disease. 4% liver and bone. We've got liver and peritoneum, lung and bone disease, and lung and pleura. So we're looking at, again, tracking right along with what's in the literature. And if we look at people who have local disease at baseline, liver being the largest segment of people, lung being the next largest segment, and I'll bet if we peel back this other segment, that's going to be bone presentation in a lot of smaller segments too. If you're in the registry, thank you so much for participating. You'll get 6-month surveys. Those 6-month surveys are important to tell us how or if your EHE tumors have changed, if you have new tumors, if you have growing tumors, or you have tumors in a new organ. It's important for you to tell us if you've started a new treatment, or very importantly, if you stopped a new treatment. Why did you stop that treatment? Again, this is in your own words. Did that treatment work for you? Did it improve your signs and symptoms, or did it improve your tumors? So thank you for participating. Thank you if you'll consider joining the registry and contributing to this initiative. So the next project that I want to talk about is the EHE Biobank, and this is a project that is significantly advancing EHE science. Through tissue donation, people can donate their tumor tissue, and that directly allows researchers to develop living cell lines and other disease models that can be used to identify treatments and eventually a cure for EHE. So this is a donation that has a direct impact. Now, tissue donation, the tissue that's donated can be used for other studies. We can look at genetics and proteomics of EHE, but importantly, we are really focused right now on model development because we really need more models in order to advance drug development. So I encourage you, if you have EHE, to make a plan to donate your tissue. If you're considering surgery, or a transplant perhaps, and you don't have to have a surgery scheduled. You don't even have to have made a decision to have surgery or a transplant. If you're even thinking about these things, please make a plan ahead, You can contact us if you have, maybe you have accumulating fluid in your lung or pleural cavity or in your abdomen, and the draining of that fluid could eliminate symptoms over time. If you're talking with your doctor about that and you know that you have these presentations of disease, that fluid being drained will otherwise be poured down the drain and thrown away, and it could be rich to study EHE. So please talk with us about that. And another way that you can contribute to EHE science is by leaving a legacy. Postmortem collection is really important. It can be significant to advance EHE science. I encourage you to call Patty Cogswell. Patty is our EHE Biobank Coordinator, and she can have a conversation with you to answer any questions you have. There is absolutely no obligation. You can have a call and talk with her, ask her any questions you have. I liken, if you live in the United States and you've applied for a driver's license, tissue donation, I think of a little bit of like organ donation. When you go to the Department of Motor Vehicles in the United States and you'd like to get a driver's license or other identification, one of the first questions they ask you is, would you like to be an organ donor? In the event that something untoward happens to you and your organs are healthy and then could be used to help someone else live a long life or help their disease. So tissue donation, I think of in a bit of the same way. If you have EHE tissue that isn't going to be otherwise used and it does not affect your clinical care, we encourage you to just think about making a plan to donate that tissue in advance. There are biobanks outside of the US. So the EHE biobank that I'm speaking of today specifically considers tissue that is donated in the United States, but outside of the US, you can call the EHE Rare Cancer UK, and you can also call the EHE Foundation in Australia. There are biobanks in both of those areas, and they can point you to those contacts straight away. So I want to just highlight a little bit of the progress that we've already made with the biobank. So again, this is a project that has not been open for very long. It's been open a couple of years. We have 62 participants, and that's significant. Keep in mind, a lot of EHE patients never have surgery or any procedure by which we could actually get tumor tissue or fluid. But we have had 41 specimens collected from EHE patients, and those specimens have enabled 5 EHE models in development. So we look forward to readouts from those, but look at the numbers. We have 41 specimens collected and only 5 models in development. So you can see that it takes a lot of attempts in order to get those models into development. And you can see where those specimens have come from. I've highlighted here, again, simply 46% of our donors came from surgical resections of tumors. 40% came from fluids, which are drained. And again, fluid doesn't need to be drained in the doctor's office. It could be that you're having drains at home or just at your doctor's office, and that fluid could be useful. We've had a small number of transplants. Again, that's a lot of tissue. An entire organ is being taken out. We don't need the entire organ, but it's very good to be able to study the diseased part of that organ as well as the healthy part of that organ by comparison. And then graciously, we've had several families reach out to us to donate postmortem tissue, and that is very important for advancing science. And I want to just reassure you all, biobank and all of the biobanks sit on an ethical framework. They have a legal structure with the biobank itself and any entity that we share de-identified material or data with, and the material being the tissues that we are collecting and forming models, or those tissues are under study. This all relies on the patient and clinicians to be partners in this process with us. It does have a strategic governance model. And again, if you have any questions at all, I encourage you to contact Patty Cogswell at biobank@fightehe.org, and we'll be happy to answer any questions that you have. So the last thing that I want to touch on here today are novel drugs in development. This is always exciting. We're always looking to see what's coming down the pipeline, what's going on in EHE clinical trials. And last year we talked about 2 phase 1 novel TEAD inhibitors in phase 1 clinical trials. These are still in early phase clinical trials. EHE Oncology had several report outs last year. In November of 23, they reported in a press release emerging proof of concept in EHE, which was encouraging for us to see. They reported favorable safety profile and encouraging signs of clinical activity in EHE. We were delighted to see that report, and we look forward to more readouts from them in the coming year. In December of 2023, the FDA granted orphan drug designation of IK930 to EHE, specifically for the treatment of EHE. That's important. That gives a nod to EHE and to us that there's some data there that tells them to keep going forward. You can learn more about this study, both the Novartis and the EHE trial, at the clinicaltrials.gov numbers that are listed here. I encourage you to go to these listings if you are considering a clinical trial, as a part of your care option. Please go to these clinical trials. Look at the inclusion criteria, look at where they're being conducted. I will caution you, not all of the centers that are listed on each of these trials may be enrolling EHE patients. There are other tumor types included in these studies. You might want to ask questions of your doctor, print out to your doctor and speak with them about the inclusion criteria. See if the clinical trial could be right for you. You want to make sure it's a good match if you're considering that as a clinical trial, as a care option. You're always welcome to email me at research@fighting-EHE.org. But again, I encourage you to talk with your doctor. So I just want to put a plug in. I can hardly talk with a new patient or a family. And if you have lived with EHE for quite some time or you're newly diagnosed, maybe your EHE is changing. You may have new signs or symptoms. Perhaps you have a new organ involvement. I encourage you to read this publication. It also warrants rereading from time to time. It's the consensus paper from the community of experts on EHE. It is our best single source to point you to learn more about this disease and get informed. Read this, share it with those who you care about and who you trust to help you in your care journey. And if you're not seeing a doctor at a major academic medical center, or perhaps you're not seeing someone who knows anything or very much about EHE because it is an ultra rare cancer, take this to your doctor. You can find a patient-friendly version on our website, and then there's a clinical version that's a deeper read for your doctor. Go to fighting-EHE.org and learn more and download this paper. So I just want to finish today with saying thank you so much. All of this, everything that I've talked about today, it all starts with you. It's only possible really because of you. Maybe you supported research at some point in your life, in your journey with someone else who has EHE, or if you have EHE, your participation in trials and other types of research, or maybe it was your financial donation. However you have done this, everything we've talked about today is only possible because of you, and truly all of this is done for you. So thank you very much for your contributions. Thank you for being here today, and I look forward to answering your questions. So Maggie, I'll turn it back over to you for questions and discussion.
Sure. First of all, thank you. That was a fantastic presentation. So much information. We do have some questions. Okay, I'm going to start with regarding the EHE Biobank. Someone has asked, can you submit tissue from a biopsy?
So we generally do not take tissue from a biopsy, particularly a needle biopsy or cord needle biopsy, because there's often insufficient tissue to work with, and we do not want to compromise clinical care. We would leave that sample with the clinician in order for diagnostics that the patient needs. If you're having a full tumor resected and that tissue is then going to be biopsied, then yes, we would be interested in that. So perhaps you already have EHE and you're going to have a biopsy of another organ to see if that tumor is EHE. That would be very important for us. I can say that learning from Hugh Leonard in the UK. The EHE Biobank in the UK does take biopsy samples. So if you're having a biopsy done there, you might want to contact the UK Biobank to learn more about biopsy samples that they would take. And that is, again, specific really just to the UK.
Okay. Okay, and then we have another question that if you've had a procedure and you've donated tissue to the EHE Biobank, can you donate again if you're having a second procedure or another procedure?
Absolutely. And that's a great question because we would very much like to get a second sample from you, even if you have donated before. You could have donated a tumor sample from a different organ. You might have donated fluid if you had fluid drained, and we would be very, very eager to get subsequent or second fluid drains and others if you have those. But if you've had tissue taken out of your body, and then let's say years later you want to have another biopsy or another resection, you need to have another tumor taken out. We also want to look at that tissue to see, did your EHE change? Did you develop any new mutations or have any other translocations that could be targetable? So it's very important to study that. So yes, the answer is yes.
Okay. Okay, the next question gets back to the new X Cures project. An attendee is asking, what about old patient records, like as old as 2006? Can those be incorporated into that?
Absolutely. So I will get with you separately, but the short answer is yes. Any record that is on file that we can gain from the hospital, we can use that. So very, very old records. And that project also seeks to keep going forward. So it would look back at your full medical history from your diagnosis of EHE forward. So yeah.
Okay. And then another person has asked, what about MRIs and CTs? Are those types of things incorporated in these observational studies?
So the XCures prospective study will collect those data because they are part of your comprehensive medical records. From the registry standpoint and the biobank, we are not actually collecting those records because we don't need them as part of that. But yes, those data would get collected, particularly the reports. So your radiological report from the scans that you've had would be looked at. And then further, we'd hope to study the actual image itself.
Okay. All right. I think that's all the questions that we had in the chat. But I do want to say, if you think of something, Denise is always available. You can reach her with those kinds of questions at research@fightehe.org. Well, I thank you, Denise. Again, that was a really, really interesting presentation. And I want to thank everyone who attended, for taking the time out of your Saturday for coming. We appreciate it for sure. And if you have any more if you want to do a deeper dive on any of this research, if you go to the research tab of our website and kind of go through everything that's under there, you'll see a lot of what Denise was talking about today. So I encourage you to do that. If you have not signed up for our email newsletter, you should do that. That's also on the very front page of our website. You can subscribe to it there. We send out a monthly newsletter. We often put research information in there as well. Let me think. I think that's all I have to plug right now. Again, thank you for your time. This was a wonderful presentation. Denise, any closing sentiments for us?
Thank you all for coming. We really, really appreciate it. I encourage you, please send me questions if you have them. I'll be glad to answer them anytime at research@fightehe.org. Thanks for your time and attention today, and I look forward to seeing you again.